Hello, I'm Morgan McClenny, and today I'm going to talk about how specific signals inside our cells enable pancreatic cancer to grow. Using a research study that analyzed three important proteins, ERK, IKKε, and TBK1. The general principle is straightforward, despite the name seeming complex. Some internal signals within the cell become locked in the on state, which contributes to the growth of cancer. Understanding which signals are turned on can assist researchers into figuring out how to slow the cancer down. First, for some background on pancreatic cancer. Pancreatic cancer is one of the most dangerous cancers because it often spreads before it's identified. The chemical signals within the cancer cells encourage them to proliferate, divide, and spread throughout the whole body, which is one of the reasons why it's able to spread so quickly. These signals typically assist healthy cells in responding to stress or repairing damage. But with cancer, the same signals can become overactive and start telling the cells to grow when they don't need to. Because of this, scientists investigate these signals to understand what makes pancreatic cancer so aggressive. The work I'm presenting today focuses on three proteins that operate like messengers. The first is ERK, which sends a grow faster message. The second is IKKε, a protein that helps cancer cells survive and migrate. And the third is TBK1, which enhances the effects of IKKε and works closely with it. The first question posed by the researchers was whether ERK aids in activating IKKε and whether this cooperation increases the aggressiveness of pancreatic cancer. To answer this question, the researchers undertook many tests. First, they suppressed ERK inside pancreatic cancer cells and measured what happened to IKKε. They employed a method known as Western blot, which is essentially a means of quantifying the amount of a certain protein within a cell. Additionally, they measured the cancer cell's growth rate and mobility, two really important characteristics for the spread of cancer. Lastly, they used statistical tests to examine the data to ensure that the outcomes weren't merely coincidental. On this slide, I'll highlight two key findings that clarify the significance of these proteins in pancreatic cancer, ERK, IKKε, and TBK1. The first illustration on the left contrasts normal pancreas tissue versus pancreatic tumor tissue. What the researchers found is that tumors have much higher levels of the proteins IKKe and TBK1, and not only are they present in higher amounts, but they're also switched on. The tumors also show higher activity of ERK, which is expected because many pancreatic cancers have a mutation that keeps ERK turned on all the time. All of this indicates that these signals are necessary for the growth of malignant tumors. The second figure on the right shows why this is crucial. When scientists utilized a technology to turn off TBK1 or IKKe in cancer cells, the cells grew considerably more slowly and created far fewer colonies. When both proteins were switched off at the same time, the cells barely expanded at all, and many of them actually died. This indicates that these proteins are not only found in cancer cells, but are essential to their survival. This data suggests that these signaling proteins are helping pancreatic cancer grow and stay alive, which is why they are essential targets for investigation. These results are significant because they imply that inhibiting ERK may impair cancer cells in a variety of ways. In addition to blocking ERK's own growth signal, it would also lower IKKe levels, depriving cancer of one of its survival strategies. A potential next step for the study is to examine whether suppressing both ERK and TBK1 at the same time could halt the cancer even more efficiently. The main conclusion is that these internal signals contribute to the growth of pancreatic cancer, and by understanding them, researchers can develop more efficient treatments. Thank you for your time.